Why don't you start trying to screen share?
Yes, just give me a moment.
Ooh, another great cover image.
Yes, hopefully one day. So yes, thank you again for having me here today, and thank you so much everyone for joining in today. I'm Elisabeth Kugler. I'm a PhD student at the University of Sheffield, and I'm even closer to defending. It's 8 days, so 12th of June is the day of days for me. So today I'm going to talk about the endothelial cell membrane behavior called Kugon. So in our lab, as you can already appreciate from this image, we're using zebrafish to study cardiovascular development and disease. And there's several reasons for us to do so. Firstly, as a human, we share about 70% of our genes with zebrafish, allowing us to really study a lot of processes in fish and translate them back towards human. But also, fish develop very rapidly ex utero, and they are embryonically transparent, as you can see here. So this is a 4-day-old fish where you look at the side view. It would be the eye, the brain sits here, the heart sits here, this is the egg yolk, and this is the trunk. And because they're so transparent, we can really look inside their bodies. But also, we can produce transgenic lines, like you can see here, to really visualize different tissues of interest with incredibly high specificity. And again, this is the same fish, but now visualizing the vasculature. And here on the bottom is the dorsal view, so the back of the head. So having these transgenic lines in light sheet microscopy, we can really acquire data about the vasculature in a very great anatomical depth, and this is shown here, where we have the 3D rendering of the head vasculature in a 3-day-old fish. So now we look at the ventral section, basically, which is the basilar artery. We look at the left eye, now it's like the lateral view. And now we go to the dorsal part, and so here would be the midbrain, and here would be the hindbrain. And so this whole section is about 700 micrometers in width and about 400 micrometers in depth. And this is just to visualize really that we can acquire all these data in the whole cerebral vasculature at a very great resolution. But also, we're really able to study very fast and dynamic processes, which is shown here, where we look at cardiac contraction. So now we look at the ventral view of the fish. And so these here are the gills, this is the heart, and here's basically the duct of Cuvier leading the red blood cells into the heart. It's just color-coded by depth, so we can just preserve basically the information of position in these animals. But also, we can study data over very long periods of time, which is great because of the very low phototoxicity in light sheet microscopy. And this is shown here in a time lapse over 21 hours from 2 to 3 days. And we look at the side view of this fish now, whereas here's the brain, so this is the eye section, the gills, the heart, again, the duct of Cuvier, and down here we have the subintestinal vein. And because we're able to really study or acquire these data over such long periods of time, we can really follow anastomosis, remodeling, luminization, and all these intricate processes with an incredibly high detail. But at the moment, there's no quantification pipeline to really assess, for example, how vascular lumen, network length, branching points, or anything like this is changing during development, or after genetic manipulation, or after treatment of drugs. And this is what my actual PhD project is about, namely to produce a quantification pipeline to assess the vasculature in the zebrafish in the head in 3 dimensions. And this is really starting off with understanding your data properties in terms of signal, noise, artifacts, etc., and how you can improve your data so you have an enhanced and better image quality for the following quantifications and segmentation. Because segmentation is the step where you basically binarize your vessels, and then you can quantify your vessel volume, your diameters, etc., and build on this by extracting your network and really identifying the skeleton properties in terms of complexity, branching points, etc., etc. But all this is great, and so we want to take it a step further and really compare different embryos to each other. And we can do this by something called image registration, which is bringing different embryos into one spatial coordinate system. And here it's just showing 6 fish being overlapped on top of each other. And if we average these fish out, we then can extract really regions of similarity shown in yellow and in red, and regions of variability in blue. But today I don't want to talk about this, but about a side project in my PhD, which was about the discovery and characterization of a new endothelial cell membrane behavior, or previously undescribed cell membrane behavior. And so in 2017, when I was looking at the data for my actual kind of quantification project, I was observing these protrusions as shown here with the black arrowheads. And this is what they look in 2D, but in 3D, they actually look like this. So this here is the structure of interest, and underneath is just a vessel segment. And because they are spherical in shape in 3 dimensions, we have termed them Kugeln, which is the German word for sphere. And so we really wanted to know a little bit more about these structures. And first we looked at time-lapse data. And so this is a time-lapse over about half an hour in the midbrain of the zebrafish, and all the different arrowheads indicate coolworms. So it's an example which has quite a lot of them. But I'd like to really focus on this one here, which shows probably the most interesting behavior, because it protrudes outwards from the parent cells, it has an oscillatory shape change, and then it goes back into the parent cell. So about now you should see this oscillation and then the retraction back into the parent cell. And these data really showed us that coolworms are very dynamic, so they can change their shape, but also that they're transient. So they always retract back into the parent cell. They never burst, shed, or anastomose from the vessels. And so we wanted to know where we would find coolworm. And we were actually quite surprised by finding that they're only found in the head vasculature and never in the trunk or any other vascular beds. And they're especially found in the midbrain vessels, shown here in green. So we have about 60% of coolworm here, and about 70% of coolworm protrude from the hindbrain vessels. And only very, very few are found in more peripheral vessels. And also we found that coolworm are only robustly formed over 3 days post-fertilization. So you would never be able to see them in a one-day-old fish, or at least we haven't. And so we next wanted to know a little bit more about the signaling pathways, which do play a role in coolworm formation. And the kind of two most obvious candidates were VEGF and Notch signaling. So VEGF signaling you can be inhibited by a drug called AB951. And if we do this, we see that there is actually a significant increase in coolworm number, suggesting that VEGF actually inhibits their formation. While on the other hand, if we inhibit Notch signaling with a drug called DAPT, we actually have a significant loss of them. And so this showed that Notch signaling is needed for their formation. And this is really interesting, and we're really trying to decipher now which individual ligands or receptors do play a role in this. But this was really quite surprising and very, very beautiful to find. And so moving on from this, we then wanted to know a little bit more about their cell biology. And in zebrafish, as I said, we have a variety of different transgenics available to really study the different subcellular compartments of endothelial cells. And so these three down here are three different transgenic line constructs where we have the fluorophore being tagged to the membrane. So they're membrane-specific transgenic reporter lines. And for all three of these, we do see coolworm forming. But if we look in a non-membrane specific transgenic like these here, so this is actin reporter and this is cytosol reporter, we're not able to see coolworm. So these data really showed us that coolworm are membrane-specific protrusions, and this is hugely interesting to us, and we're really looking into this far, far more now. But obviously we then wanted to know if coolworm were similar or dissimilar to anything previously described. And two obvious candidates for us to look at were cellular blebs and aneurysms. So cell blebs are normally formed by individual cells and they're cytosol-driven, and aneurysms are formed by multiple cells and they're blood-filled. And so the first step to do was to look at cytosol in combination with the vascular membrane. So we have a double transgenic now where we have the cytosol in green and the vascular membrane is shown in magenta. And so what we do see is that there's actually no cytosol inside coolworm, showing that cytosol is not the driving factor of coolworm formation, and therefore they're different to cellular blebs. And so we then next looked at endothelial cell nuclei, again with the same principle that we have green nuclei and magenta membrane. And we don't see any nuclei forming or inside coolworm, and we have never observed any in them, suggesting that coolworm are actually formed by individual cells and therefore different to aneurysms. And this was actually confirmed when we looked at blood flow. And so we can do this by injecting dextran into the bloodstream. And so we see here a filled blood lumen, but there is no dextran inside coolworm. And this, together with various other experiments, really showed us that blood flow is not the driving factor of coolworm formation, and therefore they're really different to aneurysms. But what we did find in this Emerald Reports paper was actually that about 60% of coolworm are filled with nitric oxide, so they turn green upon contact with DAF-M, which is a live dye for nitric oxide. And so we were really intrigued to learn a little bit more about this nitric oxide, and this is really work that has been done now after this publication, because obviously nitric oxide is known to be associated with other signaling pathways, and we thought, okay, maybe coolworm function as a storage of this nitric oxide. So what we did was to supply a drug called SMP, or sodium nitroprusside, and what we found is actually that there's a significant reduction of the number of coolworm if we supply SMP. So our current thought process is that if coolworm are a storage for nitric oxide, maybe this artificial supply with SMP is actually kind of making coolworm dispensable. And so this is kind of one way of thought that we're kind of now examining a little bit more. We're especially also interested into learning about the different upstream factors such as KLF2s or lovastatin in blood flow, and how these really impact this nitric oxide inside Coolon. Then we obviously also want to look at the actual function of nitric oxide as well as the actual function of Coolon. And for this, we go to the cellular level and the biomechanics. And since our publication, we have learned that Coolon are actually formed by individual cells, and we can see this here, where we have claudin visualizing tight junctions. So this is the right junction and left junction, and one endothelial cell in the center here. And this allows us then to approximate the surface or the structure of Coolon as well as their parent cells. And if we quantify, for example, the surface for this particular Kugel, we see that the surface is about 400 micrometers squared, whereas here for the parent cell, it's about 270. So the surface of Kugel can actually be far larger than the surface of their parent cells, which is hugely interesting because that raised the question, how are Kugel formed, how are they being made stable, and how does it happen that they don't rip their parent cells apart? And so now I'm really wanting to look into an image-based mathematical model of really describing these forces of the membrane, shown here in magenta, together with the actin cortex and the different linker proteins, as well as cytosol and the lumen, kind of the pressure of the lumen, as we know that endothelial cells unsheath the lumen. And so this is really very much still in work in progress, but we're really interested into learning this association of the membrane together with the actin cortex, because we have previously shown that the actin, shown here in green, is underlying the membrane of the Kugel dome, but especially it has a very high enrichment at the neck of Kugel formation, after Kugel formation. And so now we really want to know how these both, so the membrane and the actin, really form together and how they kind of dynamically interact during the lifetime of a Kugel. And for this, we very much go kind of back to the start, namely using biomedical image analysis. And we started just recently now to really examining how to segment and automatically really characterize basically the structure of Kugel. And so with the segmentation, we're hopefully able to very soon quantify the surface, their volume, their curvature, etc., and then examine this over the lifetime of a Kugel together with the actin cortex. But like I said, this is still very much work in progress. So with this, I'd just like to very briefly summarize. We have learned that Kugel are transient and dynamic structures, that they're specific to the brain and only over 3 days post-fertilization. And we know that they need notch signaling, as VEGF actually inhibits them, but we still don't know the really intricate or the exact ligands and receptors. So this is future work, definitely. But also at the moment, I'm very much looking into the role of nitric oxide, and with being at home with COVID, a lot of computational analysis is getting done. So I'm looking into the biomechanics of Kugel at the moment. And so this is still work in progress. And so with this, I'd really like to thank a lot of people involved in all the different projects throughout my PhD, but especially my supervisors, Paul Armitage and Tim Chico. And in the Kugel project, it was really Stefan Schulte-Marker, Jan Husken, Rob Wilkinson, Ryan McDonald, and many, many more who really supported this work. And I'd also like to thank my funders from the University of Sheffield, especially from the department IICD, as well as Insignia Institute for really supporting my work. And with this, I'd like to thank all of you for joining in, and I'm really happy to take any questions.
Ooh, another great talk.
Thank you.
That is the prerequisite to like, that's so crazy that both of you guys are going to defend within a couple of days.
Yeah. Especially in these times, it's a bit stressful, I guess, but can't help it, right?
True. So we have some questions. Okay, but I have to ask, like, that's also really similar to your last name.
Yeah, so I am Austrian actually.
Okay.
So they're Austrian chocolates, which are round. They're called Mozart Kugeln. And my boss was eating those, and then he was like, "Oh, Kugel means sphere," and so we called them spheres. And now it's like, okay, fine, Kugel.
When I saw that, I was like, did she name it after herself? Which is like a thing.
Unfortunately, not.
To do.
But
I think you could go with that story.
I know. All right, I'm seeing the questions now. Mike, thank you so much for your question. Cardiac stress, so that's actually something that I want to look at as soon as the lab's open. So in fish, we can increase and decrease, for example, shear stress very easily by injecting morpholinos or EPO mRNA. When we actually change the cardiac stress by, for example, temperature changes, that had no effect. Similarly, when we stop the heart, for example, with anesthetics, that itself had no effect. But obviously, like the long-term effect of blood shear stress is something that I want to look at, especially with respect to the nitric oxide story. So thank you very much for your question. And Becca, why was notch signaling, or why was notch a candidate for you to examine in your work? So in angiogenesis, the main two pathways which regulate angiogenesis are VEGF and notch. So VEGF is the vascular endothelial growth factor, which is a very pro-angiogenic pathway and is pro-proliferation and very productive, basically, whereas notch signaling is more counterbalancing and anti-proliferation. So it's more really like setting or like a counterbalance to VEGF. And in our lab, it was just like the first thought really to say, okay, VEGF, notch, just, you know, apply it and see what happens. Because especially in fish, it's very simple to just add a drug to water and then it can inhibit or decrease.
True. That is really nice.
It's very, very easy to do these experiments. Raga, thank you for the question. Will it be possible to use a FLIM dye or membrane resident protein to look at biomechanics via light sheet? So yes, I am. So my kind of biomechanics work has just really recently started before the lockdown. So it's very unfortunate that I don't have more data, obviously, but I'd like to really look a little bit more into this. And for example, perform FRAP experiments, etc. But the issue with really, for example, a FRAP or FLIM experiment, or also like laser injury, is that these cool are actually quite deep into the brain tissue. And so we kind of hold off from that because it might just injure the brain. And so we don't really know if that is then kind of a direct or indirect impact of cool formation, or like of this interference with the laser, basically. Right. So we're now thinking about 3D co-culturing and maybe then seeing what happens. We might be able to induce cool, and then life would be so much easier.
I actually have one additional question from someone watching on YouTube. Amazing. Says, all zebrafish spontaneous larval bleeding mutations primarily exhibit hemorrhage in the brain. Have you thought of looking whether there's any correlation with Kuglin in these mutants?
They can also be drug induced, and I've actually done some preliminary experiments in, I think, February, just before the lab lockdown. Unfortunately, I had a too low drug dose to see anything, so those experiments have to be repeated, because I'm really very much interested to obviously see what happens if we have spontaneous hemorrhaging. Right. And similarly, I'd also like to see what happens if we induce hemorrhaging with injury, for example, just in one brain hemisphere. So that's kind of, as soon as we can go back to the lab, all these experiments will be wonderful to get data from.
Oh, great. Okay, thank you so much.
Thank you so much for having me.
Good questions, everyone. All right, we are moving on to our last, but certainly not least,